glad you're here. So only people who's allowed to do the remote learnings with excused absence. Okay, you have to provide document. You stamp on it. Oh, it's verified. Yes. Okay. So in general, so you do need to attend the class. Yeah. Okay. So I'm glad you're here. Just need to keep coming back. Okay. So hopefully some of you guys, I see you already submitted the discussions, right? So microbiology 1 and microbiology 2, the weekly assignment is kind of similar. What do you do weekly? Lobster, yes. We continue to do the lobster, yes. We also do what? Discussion, yes. We also do what? Yes, discussion, you need to submit total 3 posts, right? The first one is your, any question you guys have? You want to discuss the question here? No? Oh, okay. Texas. Okay, so we can do that outside class, yes? Okay, awesome. Yeah. So we do the lecture, virtual lab help you, right? For example, the lymphatic organs. This week this week we're going to first start looking at the immune system, okay? Anatomy of immune system. And then weekly quiz. We have weekly quiz too, right? So how many times you need to try the weekly quiz? And yes, many times as you can. And also another thing is with the weekly quiz, right? It's open book or closed book. It's open book, but towards the end of the week, on Sunday, you say, oh, I've done this week's learning. I'm on wrap up this week's learning, get ready to move on to next week's learning. What you need to do? You might want to try closed book again. Just make sure you remember. Maybe the open book, oh, I get 100% the open book. Do you remember them? Right? If they ask the question in different ways, can you still answer them? Yes. So that's so if you do the closed book at 10, hey, open book 100%, closed book 20%. Guess what? Well, are you done with that week's learning? No, right? So if you get activities, so what you can do? What you can do, right? So you can go to the weekly lesson plan, right? You can do some reading, right? For example, I have the link of the chapter you need to read. Yes, you can go back and read that section. Oh, this is the question I get wrong. This section I need to read. You don't have to read the whole chapter. You could just read the select section that you have trouble with. Yes. Okay. And you can also watch all these videos. Yes, right here. I have 2 textbooks. So besides the normal microbiology textbook, so this one's anatomy textbook, free. Right? So you just read the whole chapters. Because in this class, we also going to read. Remember last quarter we learned the innate immune response. First line defense, second line defense. Right? So in this class, we're going to learn the third line defense. The third line defense involves T cell, B cell, antibody, involves bone marrow, thymus gland, lymph node, all that stuff. So I have a textbook for anatomy in here for you to read for this week. Yes, comedian, yeah, right? Okay, so we have video to watch, we have book to read, right? In addition, what else you could do? What else you could do? Could you draw it to know it? Yes, draw it to know it, right? So if you're going to draw it to know it, I build a study plan for different class. Which study plan we're going to use? Everyone look at here. Which study plan you're going to choose? Microbiology 2, okay. All right. Then we're in the microbiology 2. So if you look at the section, right? Let's close this one up. You see the sections, right? Has cell, genetics, hematopoiesis, the cells in the blood, and immune response. Right? So for this week, for example, draw to know it, I'm going to click here. Right? Click here. You see different type of cells. So today we're going to look at the organs of the immune system. Spleen, thymus, lymph node right here. Right? Down the road, we're going to learn from have cell function, antibody function, cell-mediated immunity, tumor immunity right here. See? Right? So this week, for today, for example, you can do this. See, all these resources to help you to do what? To understand the topic. So first section of this class before the midterm exam, maybe it's immunology. Right? Immunology, if you understand it, it's fun and easy. If you don't understand it, it's hard. That makes sense? You really focus on, I know, if you just redo the quizzes, you see all the questions. But if you don't understand it, you just see 100 questions in week 1. Each question is separate. They don't have connection. That's a lot of remembering. But if you understand it, then you have 20 questions about phagocytosis. If you understand phagocytosis, you can answer all these 20 questions. That makes sense? You have 10 questions about T cell, formation, activation. If you understand the process, you can answer all these questions. You see, have to understand. Okay, so this interactive, besides the virtual lab, besides the tune to know it, besides reading the book, hopefully besides watching animation videos, you truly understand. Then you can apply and it doesn't matter which question we ask. Make sense? Okay, so first part of this class literally is immunology. So you have to focus on understanding. Okay? So that's the resources. Yeah? Now, so this is a continuation of microbiology 1, but I made it more fun. I don't want to feel like I'm taking the same class, just boring class, right? We have fun activity. One of the fun activity we're going to do tomorrow is Kahoot game. So tomorrow we're going to do Kahoot game. And another fun activity is what? We have new effort grade. We have this literature review, microbial infection literature review. You can practice not only your reading skill, but your writing skill. Yeah. Right. So this is due by week 9, so you have time, but you can start looking at this, thinking about which disease you like, and this is rubrics in there. Right? How do you find this information? You see, right? Microbial disease literature review. You like it? Thank you so much. Yeah. See, it's fun, right? So that's it. This class is a little bit different from last class, so we have fun. Okay. So the grade is very similar. So the discussion, quiz, literature review, and lecture all part of your effort grade. Try to finish them before the final exam, before the weekly due date. That's truly learning. If you did not do it after final, I get F. Now I do it, then that not help you his final exam. You see, you can pass. Yeah, right. So you do it and follow the weekly schedule. Okay. So that's the exam will be midterm final exam. It's a major exam. So if you, in general, if you cannot pass the midterm final exam, probably not gonna pass class. Yeah, just like right. So you have to make a, yes. Okay, awesome. So that's the introduction of the course. Any question? No? Okay. If you don't have questions, so if you the organization of this class, like what's in the PowerPoint file, you just go to week 1, right? You can see the lectures. It's 3 files. Today we're going to look at the organ of the lymphatic system, cell of the lymphatic system. Tomorrow we're going to look at how do the cell communicate with each other. Then tomorrow we're going to have. Today we're going to have only check. Tomorrow we're going to do cooking. Okay. So. You guys ready tomorrow? So first of all, we can look at the organs of the lymphatic system. Okay. So organs of lymphatic system. So I have a lot of pictures in my PowerPoint slides. So because I think a picture tell better story. Yes, we have reading. We also have pictures. Okay. So obviously this is human, right? Based on the function, the organ of the lymphatic system grouped into two types. One is primary lymphoid organ, another one is secondary lymphoid organ. Yes. So based on the picture, can you tell me which organ are the primary lymphoid organ? Everyone, which one? Bone marrow and thymus. Okay. And then, based on this picture, can you tell me the example of secondary lymphoid organ? Lymph nodes, spleen, tonsils, adenoids, right? What else? Mesenteric lymph nodes, Peyer's patch, right? Urogenital lymph node, you get an idea. Cervical lymph node, yes? Okay. So now we sort it out. What's that? Yes. So just like in the cardiovascular system, right? You have blood vessels connecting all this, right? Deliver the nutrients, they transport waste, all that stuff. Now, in the what's connecting all this lymphatic tissue? How do they get antigen? Through what? Lymphatic vessels. Lymphatic vessels. Yes? Okay. So the lymphatic system, the structure include what? Primary lymphoid organ, secondary lymphoid organ, and lymphatic vessels. Yes? Okay. So now we're going to show you each one. Okay? Bone marrows. Where do you find your bone marrow? Right? So which type of bone marrow contains the stem cell? Red or yellow? Red bone marrow, right? So red bone marrow contains stem cell. So the stem cell in red bone marrow generate all the cells in your blood. All the cells in your blood come from where? Red bone marrow. Okay. So if you look at the picture, right? So if you look at between children and adults, so the children, all the bone marrows are red. Yes. For adults, you can see adult, the red bone marrow maybe is finding where? In the flat bones or tip of long bones. Yes. Okay. Now, the stem cell, we call them hematopoietic stem cell. So hematopoietic tissue, hematopoietic stem cell, talking about this cell able to generate all kinds of cells in blood. Red blood cell, variety of white blood cells, platelet, all come from what? Hematopoietic stem cell in the red bone. Got it, everyone? Okay, awesome. See, relatively easy, yes? So now we're talking about, hey, the hematopoietic stem cell in red bone marrow able to become all kinds of cells in your blood. How do they know which type of cell they need to become? Right? How do they know the host needs a red blood cell or neutrophil or eosinophil? Yes? How do they know? Through what? Through what? Growth hormones, right? Growth factors. So this is the chemicals. Now look at the table. Look at the table, right? The table is talking about class 1 cytokines, right? So class 1 cytokines include the chemicals. They are the one guide the hematopoietic stem cell in the red bone marrow to become which type of cell. They're going to tell the hematopoietic stem cell, hey, we need more T cell, we need more red blood cell. So some of these cytokines are hormones. For example, erythropoietin. Have you heard of EPO, erythropoietin? Right? That's a hormone talking to the red bone marrow hematopoietic stem cell. Hey, we need more red blood cell. Right? If you need more white blood cell, guess what? It's interleukin 2, for example, 3, 4. Hey, we need variety of white blood cell. You see? So the cytokines and the hormones, the growth factors, they're the one telling the cells in red bone marrow, hey, we need more what type of cell based on host condition. Make sense? Okay. So now, how do they talk into the, so these cytokines, these small proteins, the growth hormones, how do they talk to this hematopoietic stem cell? How do they talk? Through what? Interaction with the surface receptor. Where do you find the surface receptor? Surface receptor on the cell membrane of hematopoietic stem cell. So the cell membrane of hematopoietic stem cell have a receptor talking to different signals. Depending on which signal binds to receptor, then they're going to turn on, go to nucleus, turn on, say, hey, we need more red blood cell, neutrophil, the T cell, B cell, this, that. Make sense now? Follow so far? Right? Okay. Follow? Yes? Okay. Now, another thing is hematopoietic stem cell also do the self-renew. Means they will generate more different type of cell, but then also make sure they have a reserve for themselves. So they're going to, some cell go through mitosis, some cell you're going to keep as a hematopoietic stem cell. Some cells become activated, become different type of cell in the blood. Make sense, everyone? Okay. So now, that's the process. Now we're going to look at over here. Oh, yeah, this is a good picture. Okay. So this picture is talking about, just show you the step. So it's like, the hematopoietic stem cell in a red bone marrow, for example, generate white blood cell, neutrophil, macrophage. It's one step or it's multiple steps. This picture show you what? Most cell in the blood coming from hematopoietic stem cell through what? Multiple steps. Yes, multiple steps. So each step they have tightly regulated by different type of hormone. If you look at this picture, right? You guys follow. So beginning, that's the hematopoietic stem cell in red bone marrow. Some of the cells renew, and then they start differentiating. So first two big differentiations what? Myeloid progenitor and lymphoid progenitor. So lymphoid progenitor will give off what? Lymphoid progenitor. Progenitor is the ancestor of the something, right? The English progenitor means what? The precursor of something, right? So lymphoid progenitor is a precursor cell for what? Lymphocytes. Lymphocytes. Make sense? Right? Who are the lymphocytes? The lymphocytes, you can see T cell, B cell, natural killer cell, right? They are the lymphocytes. They come from what? Lymphoid progenitor. Make sense? Follow so far? And then another big branch called what? Myeloid progenitor. Yes? Myeloid progenitor. So this group, this one will give off what type cell? Platelet, red blood cell, including granulocyte, eosinophil, neutrophil, basophil, this are. And both of this group also give off what? Dendritic cells. Got it? So now I'm familiar with this table, right? Or this diagram. Now we're going to use this one to answer the next couple questions to see. Ready? So this is the key for this question. Just check. So this is a question, you go back to the slide and follow the diagram, find out which lineage give off macrophage. You know what I'm doing, right? That's easy. Anyone find it? So somebody said F. You guys agree F? Yeah. F. F. Agree F? Do we need to go back or you guys all sorted out? You did, right? F. Okay, F is correct also. Good job. So now let's continue to the next question. See? So again, we use this slide, right? To sort out this one. Dendritic cell, right? Dendritic cell. Let's look at dendritic cell. So dendritic cell, right? Dendritic cell. Yes. Right? Look at dendritic cell here and here. Yes. Then come back here. What's the best answer?
D.
Dendritic cell. What's the best answer?
G.
You see, dendritic cell could come from here, right? Dendritic cell also can come from here, right? Yes? So best answer is what? Dendritic cell is all of this. Yes? Yeah. All of this. So look, left side, you see dendritic cell over here? See? Lymphoid progenitor give off what? Dendritic cell. This comes from hematopoietic cell. Dendritic cell also come from myeloid progenitor. Also come from what? Monocytes. Yeah. Yeah, see? Right? So best answer is what? All of this. All of this. So now you see, if you want to know why dendritic cell, no matter where you're going to give off dendritic cell, means the dendritic cell must be very important, you see? So now I'm going to tell you the function of dendritic cell. You can write down it's called what? Antigen. Know how to write antigen? Antigen presentation. Present antigen. So what's the function of dendritic cell? Antigen presentation. They are the professional antigen presentation. The professional means that the only thing they do is present antigen. Antigen presentation, dendritic cell. You see, because the presenting antigen is very important for our cells to look at it and start to figure out the strategy to get rid of it. Yes? So you see, no matter where, which lineage always give off the dendritic cell. Okay? Now, let's look at this one. Yeah, same slide. Let's sort out the T cell. These are come from where? So best answer is which one? E, right? This one easy, right? E. Okay, awesome. So now we know how to look at this slide. Yes? Okay, awesome. So this is E. Awesome. Now let's look at the structure, the primary and secondary organ, right? So now let's look at, we're going to use each one to talk about the function. So the red bone marrow, the function of red bone marrow is all the cells in the blood are originated. You know originated, right? You know English originated, yes? So all the cells in the blood are originated from what? Red bone marrow hematopoietic stem cell. Yes? And then what's the function of thymus? If you look at this picture, right? Thymus. In terms of immune system, right? In terms of immune system, red bone marrow also is the school for B cell. School for B cell means that B cell not only come from there, but also mature there. Now, the thymus is the school for T cell. When you school, you guys are familiar with school because when you're in school, you know the function of school, right? Function in school is somebody's new freshman come in, by the time you leave school, you armed with skills. Yes? And the thymus is the fresh cell, right? From the bone marrow come to the thymus. By the time they leave the thymus, they are ready to do the job as T cell. So T cell get in the thymus. Yes, question.
It's smaller as TG.
Yes. So eventually it becomes immunocompromised. Then, so when your thymus gets shrunk due to age, You're not going to have new graduates, like school shut down, and new fresh graduates. So who's doing the T cell job? Is the T cell already in where? Secondary lymph organ. So secondary lymph organ is kind of like your chiropractic office. After you graduate from Life University, hopefully you have office in the Carl Parkway. Okay, right? Carl Parkway. Oh, I have office, please come. So that office is kind of equivalent of what? Secondary lymph organ. So once the B cell, T cell finish their schooling, B cell schooling where? Bone marrow. T cell schooling where? Thymus. When they graduate, they go to secondary lymph organs. That's where they say, hey, now I'm ready to open practice. Ready to fight the microbial infection. Make sense? Okay, awesome. So now, over here, you just see how do the B cell mature in the bone marrow. So B cell mature in the bone marrow stage by stage. Stem cell, if you look at this picture, we could just analogy of hematopoietic stem cell is naked baby. How about that? Naked baby. And then how do they get educated? Hey, you need to put clothes on, right? So then you can have this pro B cell, you start to put some fabric on, pre B cell, more fabric. By the time you can like, become ready to graduate, guess what? The B cell surface have receptor. So stage by stage, naked baby means they don't have receptor on cell membrane. Then as they mature, they start to add the receptor. By the time they add all the receptors, that's likely to be very close, right? Then maybe the lab coat or maybe the professional gowns, whatever, right? Then you're ready to do the job. Sounds good? Because once you fully close, so you go out there in the blood, they look at what you wear, say, oh, I know what you do. You're a doctor, you're an engineer, you're a policeman, because you have clothes on, we can tell what you do, right? That's how the other cells see the other cell, what you do, based on what you have on cell membrane. Make sense? Yeah? Okay, awesome. So now, right? So the effector, so the mature B cell have the fully closed. And then once it becomes effector cell, they can become plasma cell, memory cell. Plasma cell, that's a cell producing antibody. Now, another thing is, you heard of autoimmune disease? Yeah, Crohn's disease, lupus, rheumatoid arthritis, type 1 diabetes, Graves' disease, Hashimoto disease. It's the name of autoimmune disease. Autoimmune disease is what? Your immune system attack yourself, right? So most time people don't have autoimmune disease later on. Most autoimmune diseases with age later on become, yeah, right? So now, so most people do not have autoimmune disease because our school in the bone marrow, thymus, have mechanism to make sure if the B cell, T cell during the schooling process, they start attack self, guess what? They get eliminated. Said, hey, if you go out there, attack yourself, you can't go out there. You have to stay here or die, right? Apoptosis, right? So what's the name of process to make sure the graduate does not go out there attack the host? Graduate T cell, B cell, called what? Negative selection. So let's answer the question. The green, the purple, right? That's the question. What's the name of the process to ensure self-tolerance? Negative selection. What's the negative selection? It's talking about, hey, if you're going to target on the self-antigen, either on the cell or on the soluble self-antigen, you can't leave. You can't leave the blood vessel. You can't go out there. Only the B cell have closed to go out there, not attack the cell, can leave the bone. Make sense? Negative selection. That's smart, yes? Right. So now, look at those pictures, right? So follicular dendritic cell helps the B cell recognize antigen, right? You look at this picture and then answer that question: which cell help naive B cell become effector cell? right? Well, effector cell is talking about plasma cell. Yes. Which cell helps the B cell become activated? You see, both have FDC. Yes. Which cell? T helper cells. Yes. TH. So T helper cell. You see, T helper cell help. Means they maybe help our cell. One of cell they help is B cell. T helper cell helps the B cell from naive stage become effector stage. Naive means fresh graduate. I'm naive. I don't know yet. And then once you see the first patient, you are not a naive chiropractic doctor. You become effector chiropractic doctor. The same thing with T cell, B cell. Once you see the antigen, you could become effector stage, not naive. Okay. So follow so far. And then the process, right? Process of B cell become a plasma cell, produce IgG. Have you heard of IgG, IgM, IgA? Different types of antibody. That we heard of, right? So antibody in your body is different forms. For now, we just know we have more than one type of antibody. Okay, you could have IgG, you could have IgM, you could have IgE, you could have IgD. Yes, you could have IgA. So right now, I'm just giving you an introduction. We can talk about this again later. Okay. So this process called what? Class switch. You switch class. Oh, I decided to produce IgA, IgD, IgM. Yes. Now the question is based on this picture. The question is which cell actually are the effector B cell producing? Which cell? Are you guys follow, right? We follow the order, yeah? Which cell actually produces antibody? Plasma cell, yes. Plasma cell. They are the one actually. So the effector B cell called what? Plasma cell. They are the one produce different type of antibody. Okay? The plasma cell. Awesome. So now next we're going to look at right, thymus. So thymus, they are primary lymph organ or secondary lymph organ? You guys still remember? Primary, yes. So what's the function of thymus? Yeah, mature. So either naive B cell or mature B cell. Okay, mature T cell or naive T cell. So thymus is a location for T cell to learn and develop into the naive T cell or mature T cell. Okay, right? If you cross section of thymus, so you see the location of thymus is where, right? So right on top of heart, where all the vessels, major vessels on top of it. But if you're not down the road, if you study anatomy, use an anatomy table or use the model, you don't see that there's a thymus on the heart bottom. Yes? Why is that? Because in the children, you have a significant gland, right? But by the time you become adult, the thymus gland shrinks so much you don't see it. Okay? And then if you cross section of thymus, you have what? Cortex and medulla. So you see the T cell gets screwed in the cortex. Once it becomes mature, then it leaves through medulla and through the blood vessel leaf. So now let's look at with all that information. Can you answer that question? So, right, strength, right? Called what? Progressive involution. So there are two glands. As you age, you shrink. One gland is the thymus gland. So that's why you connect dots, right? In the COVID-19 pandemic, which group are susceptible for severe COVID symptom or death? Seniors. Now we connect them. Yes. T cells, intracellular immunity, virus, intracellular, you have weakened the immune system against the intracellular infections such as Lyme. They connect dots. Okay. Another gland actually shrink as you age is pineal gland. Pineal gland, you guys learn pineal gland yet? Hormone, yeah, hormone regulate to the sleep. So But anyway, so let's look at the T cell, right? The schooling of the T cell in the sinus. Again, so this process is not one step. So when the cell comes from when the T cell originates from the red bone marrow, we're going to call them naked T cell, right? Or double negative. Double negative means CD4, CD negative means they don't have any clothes on, naked. Then to go into the thymus, they start to put clothes on. So you can so you go to the cortex, you become double positive. Means hey, we took two types of T cells. Right now we just introduce you to type T cells, CD4, CD8. So where do you find CD4, CD8? It's simply a protein on the surface membrane, like CD marker. It's like receptor on cell membrane. So CD4 is like, oh, the cell membrane have pink clothes. So one group, CD4 will be the cell with pink clothes. CD8 will be the cell with blue clothes. Boy and girl. Sounds good? Analogy works, right? So as they eagerly, so they all we naked, come out to the bone marrow, go to thymus, we are very eager to try any clothes. So what they put on? They put on both clothes. They put both pink and blue clothes on. So that's called a double positive, DP. See the DP? DN, DP, right? DN is double negative, DP is double positive. So DP, double positive, so we have both CD4, CD8. So they're so eager, they put both pink and blue clothes on. Say, hey, you can't do both. You have to pick. Can't be both girl and boy. Yes, you have to pick. Right? It's a gender issue, right? And anyway, right? Down the road, we learned some word from mathematics. Yes, we have both. Yeah. But right now we're talking about, so I'm using the analogy to help you understand. Then you have to pick. So that's how they pick. So in the double positive state, that's where it goes through battery test. You can't randomly just choose. You have to go through pass test. Once you pass the test, then it can become either CD4 or CD8. There's a test called double positive selection, negative selection. Okay, let's look at the test. You follow so far? Right, so in the cortex, that's where it goes through the test. Once they pass the test, they can migrate through medulla leaf. So the test, we have positive and negative selection. So you see DN double negative, DP double positive, talk about CD4, CD8. CD4, CD8 is simply just a protein on the cell membrane, like a surface marker. Okay, so how do they do the positive negative selection? Okay, so you guys still remember this question? Let's see. This one? This is talking about B cell. Yes, you guys remember this one? Yes, right? So which process ensures self-tolerance? Negative selection for B cell. And then for the T cell, same. Right? So the negative selection, you see the bottom negative selection? So the double positive, so this is the eager cell, the cell. We have all the clothes on, we're ready to do work, all you have to pick. So we have to do tests. So one of the tests is they trying to, you see the medullary cell, they can present self-antigen. So they give them self-antigen, they do the test. So if the DP double positive sinusoid, they so eagerly bind to self-antigen. They are so eager, have highest affinity. You guys know affinity, right? So if they have highest affinity, so eager, guess what? They failed which test? Negative selection. You see, you're so eager to bind self-antigen, you can't leave. Otherwise, you're going to go out there and attack self. That would be autoimmune disease. Make sense? That's negative selection, right? Over here. Now, right? So negative selection is self-tolerance, the same thing for T cell and B cell. But for T cell has additional positive selection. So what we're talking about is the T cell. If you go to read the president, so you know the president of America, right? In charge of everything, pretty much. Yes. So who's the president of the immune system? It's T helpers. T helper cell. Okay, we just use the T helper cell as or all the T cells, especially T helper cell. They suppress the immune system. So now go back to our society. So if you want to go meet the president, can you just go walk in the White House and just meet the president? Probably not. You have to go through the security check. You have to make an appointment. You have to make sure you have a record. Make sense? So the same thing in the T cell, the immune system being present, they're not going to look at antigen directly. Got it? So the B cell will directly look at antigen. Dendritic cell will directly look at antigen. Macrophage will directly look at antigen. But the T helper cell or CD4, CD8, they're not going to look at antigen directly. In case the antigen is super toxic, you kill the T cell, then your whole immune system down. You see? So how do the T cell recognize antigen? Through what? Histocompatibility complex. Everyone? Histocompatibility complex. Called what? Major histocompatibility complex, short for MHC. So now now everyone know MHC stands for what? Major histocompatibility complex. So this is the type of protein on cell membrane kind of identifies who you are. Okay? So for T cells, we think of this as a shoot, business clothes. So in order for you to go to White House, at least you need to dress up. You can't just wear the sandal and the shorts, go so on, right? You see? Or go through the interview. Yes? You have to dress business professionally. So this dendritic cell, remember we talked about dendritic cell previously? Everyone still remember? What's the function of dendritic cell? What's the function of dendritic cell? Antigen presentation. So how do we present antigen? We could think of dendritic cell as Taylor. So they're going to put the MHC business code on the antigen. So bless you. So dendritic cell grab the microorganisms. They're going to process it, and then they pick apart and put on the business suit MHC. After that, then T cell will look at it. Understand? So how this process, so how do the T cell know you have to look at antigen through the MHC model? They have to get the business codes, go through the antigen presentation. You don't look at directly. How do they do that? Through positive selection. Through positive selection. So B cell don't do that, but T cell do it. So how do they do it? Look at this, right? So you see, this is double positive sample size. So look at it out. They're not going to look at the antigen on the MHC. The MHC have business codes on, they're not looking at it. Then you go to apoptosis cell death. Yes? And then you see the middle, these two through positive selection. So they look at the antigen on the MHC that have business codes. They're looking at it. One is a low affinity, one is a high affinity. The highest one, they fail the exercise. The highest one get the eliminated. But the low affinity, this is one perfect. This one can lead as a CD4, CD8. But the high affinity still have risk for loose self-tolerance. So high affinity, they just said you can lead, but you're not going to do anything. You're just going to be a monitor, regulatory, called Treg. Natural Treg. Natural Treg come from thymus in the high affinity antigen mechanism. They said, hey, you recognize that it's not the highest one is too dangerous for autoimmune disease. You have to eliminate that. But high, they said, we're not going to ask you to do anything, but you could become RS. You know RS tax. IRS, right? Right? So the natural Treg will be like an immune system where you don't do stuff, you just regulate your monitor, make sure they don't overdo anything. Right? That's the Treg. Make sense? And the low affinity is, oh, I bind very low, but I can't recognize antigen. Then you can leave, go out there, do your job. Now we understand the positive negative selection, yes? Analogy works? Okay, awesome. So now, let's look at this question.
First A.
So first one, negative selection B cell occurs in where? In where? Negative selection of B cell occurs in where? Yeah, bone marrow, yes? Negative selection of B cell occurs in bone marrow, right? Negative selection of T cell occurs in where? Thymus. Positive selection of T cell occurs in where? Thymus. Yes? That's review, yes? The function of, yeah. Yeah, both in thymus, yes? So that's the test. They have to pass it, like you guys have to pass the board test. Part 1, part 2, yes? The T cell have to be in the thymus, pass it of negative, positive selection. Right? Okay. Now, so now let's look back over here. So this works, yes? Okay. Now we're going to look at another organ, spleen. So what's the function of spleen? Have you learned that? Have you guys learned the spleen function yet? Human anatomy. Spleen have two functions. So if you cross-section of spleen, have red pulp, white pulp. So spleen is a primary or secondary lymph organ. Secondary lymph organ. Primary is bone marrow and thymus, right? Spleen is secondary lymph organ. So spleen, the red pulp, majority red pulp, and then they have white pulp. It's like iron and the red sea, right? So the red pulp function is a graveyard for red blood cell. You know red blood cell, right? Have no nucleus. Red blood cell die routinely, yes? 120 days they die, yes? You know that? Have you learned that? Yes, okay. So where they go die? They go to spleen die. And then the white pulp, the island in Red Sea, the island, that's where the T cell, B cell graduated from the thymus bone marrow. They located the office, could be in the white pulp, the island of the Red Sea in the spleen. So if you red blood cell death, in case they have microorganisms in there, so the blood-borne pathogens, microorganisms in the blood, they can go along with that red blood cell, go to the spleen. So the white pulp will get rid of it. So white pulp, the T cell, B cell, and the white pulp, they can screening for the blood-borne pathogen. So if you have splenectomy, it's talking about if somebody having large spleen rupture, you can have to remove spleen because the bleeding can be deadly, yes? So once you remove the spleen, guess what? You have increased risk of what? Bacteremia, parasitemia, all that stuff. Make sense? Okay. All right, over here, you can see the comment like an enlarged spleen, right? Enlarged spleen. You know, basically, Epstein-Barr virus, cytomegalovirus virus, you know, cause disease in the brain. So any type of disease in the blood, multiplying in the blood, sepsis, bacterial brucellosis, typhoid fever, is microorganisms in the blood. And the malaria, Chagas disease, visceral leishmaniasis, and cortisol, eukaryotes in the blood. All that include leads to what? Enlarged spleen. Yes? Okay. Make sense? So from here, you see spleen is very important.
Epstein-Barr virus is mononucleosis, right?
Yes, infectious mononucleosis. Yes. Okay, cytomegalovirus. Yes. Right? A lot of these diseases we're going to learn again in the chapter called the cardiovascular disorder. Yes, disease and cardiovascular system. Right now, we'll just show you here. We're talking about how important spleen is. Right? Make sense? Now, next we're going to move to the lymph node. All right. Lymph node is primary or secondary lymph organ. Lymph node. Secondary, yeah, secondary lymph organ. In fact, lymph nodes are the most numerous. The most numerous secondary lymph organ is lymph node. You have lymph node, cervical lymph node, everywhere, right? Yeah, everywhere. So if you cross-section lymph node, so if you look at this, this is a drawing of lymph node, right? So you have cortex, medulla, and then you have follicles in the cortex. So the follicle over here, that's where the B cells in the germinal center, the T cells around it. So the follicles contains what? T cell, B cell. So remember the T cell, B cell pass the positive selection, right? Negative selection, right? They go to where? They could go to spleen, the white poke island in the sea, right? Red Sea, or they can go to lymph node. Where to go? Go to the follicle. Yes? And then the lymphatic drainage. Lymphatic drainage, you see the effluent coming in, they're going to filter through. So this lymph follicle. And then once they get checked, say, hey, we identify antigen, then become effector cell. Effector cell leaves. So this leads to lymphatic drainage, and also they could come in. You see the blood vessel in there as well. Right? So the function from this picture, function of the lymph node is what? Filter the lymph drainage from peripheral tissue. Yes? So the function of spleen is to filter the blood. Function of lymph nodes filter the lymph drainage from peripheral tissue. Make sense? All right, everyone? Over here, you can see the cross section. Say this is drawing. Say, hey, I'm just making it up or what, right? Over here, that's the real thing. You see the follicle, yeah, right? Histology, the medullary cortex, the follicle in the germinal center, B cell around the T cell, right? You see the T cell, B cell. See, that's real. Now we're going to use that to answer this question. So this is a question I ask you guys to look at this picture closely. Because I like this picture, tell story, yeah? Okay. So where do you find the B cells? In the cortex, right? And also specifically the cortex contains the germinal center follicle. So germinal center follicle in the cortex. The cortex have what? Follicle and the paracortex is surrounding it, right?
So A and C.
Yes, A will be a big group. The actual B cells in where? Germinal center cortex. Paracortex as T cell. See the follicle? Yes. Make sense? So if you only choose one answer, that would be A. If you have multiple answers, C could be in there as well. That makes sense? Got it? Okay. Now, if you have inflammation in lymph node, what's the symptom? Can you see it? Which doctor can see the symptom? Inflammation in lymph node. Which doctor? Oh, doctor, including which doctor you're going to become? Chiropractic doctor, see? You see the relevance? Okay. So typically in large lymph nodes, right, you can palpate it, yes? Okay. Now, what about the mucous membrane? Mucous membrane. So which organ system have mucous membrane? Right, respiratory tract, GI tract have mucous membrane. So what type of antibody in the mucous membrane? Secretory IgA dimer. Okay, secretory IgA in the mucous membrane. So the mucosal associated lymphoid tissue, most, they are primary or secondary lymphoid? Secondary. Okay. So let's look at this question. So this question is based on previous slide. So this previous slide had a lot of words, right? I want you to read it. So you guys read this information. You have M cell, you have macrophage, dendritic cell, T cell, B cell, and mucosal associated lymphoid tissue have IgA, right? Now, which one have it? That would be F. Yeah, straightforward. Okay, awesome. So the mucus and IgA, that would be part of like second line, third line defense, right? Yeah, right there. Now, Let's look at the cells, how that works. So how do you know? So this cross section, so this is drawing, right? And this is a cross section of, you see the cells up there, finger-like projection. So those cells finding where? In the gut. So gout is talking about what? Gut-associated lymphoid tissue. Gut-associated lymphoid tissue also called what? Peyer's patch. You see? You guys look at this picture, right? So you see top, the finger-like projection, V-line, micro-V-line, they increase surface area, they absorb nutrients. Then in the gut, you also heard of probiotic. You heard of probiotic? Yes? That means there's microbes right in the lumen over there. So that's why you see the follicle, you see the follicle right underneath the finger-like projection, that follicle circle, the square, the follicle. What's in the follicle? That's the office for the T cell, B cell. So you see, this drawing over here is actually sun. You see, those cells absorb nutrients from your gut. In here also have microorganisms. You could have probiotics, you could have pathogenic, E. coli, Salmonella, Shigella. Yes? So what you do is right underneath the epithelial cell, you have this follicle. In the follicle, you have T cell, B cell. Right? You see the follicle have T cell, B cell, M cell. M cell will transcytosis. So M cell will transport the sample in. And then the T cell, B cell, dendritic cell will look at it. So if there's suspicious cholera or Salmonella, E. coli, they're going to get rid of right here. Make sense? Okay, that's it. So now, so lymphatic vessel and the cells, you guys want to do it after the break? Okay, so much information, not analogy. Hopefully you hope you understand, right? Yeah. So we're gonna come back at 4:10, right? 4:10. So we're going So we look at the structure, the function of a bone marrow, the thymus, the lymph node, the spleen, yes? So the next structure we're going to look at is lymphatic vessel. So if you look at this picture, right? So you guys know the arterioles and venules, right? You see the capillary bed. So the formation of the beginning of the lymphatic vessel is where? It's near the capillary bed. So where the fluid come from? You see the fluid come from what? And we call it interstitial fluid. Interstitial fluid is the tissue fluid outside the vessels. Yes? So interstitial fluid. So some of the fluid come from the blood vessel. Yes? Right? You can see this is a capillary bed because you see the color change. Red to blue, you know why it's red to blue color? Have you learned that? Red contains what type of? Oxygenated blood. And blue vessel contains what? Venule contains what? Big oxygenated blood. So as they dumping oxygen pick up the waste, the fluid also in the tissue fluid. So some of tissue fluid will drain into what? Lymphatic vessel. Lymphatic vessel collect tissue fluid, including if there's a microbial infection in the tissue, guess what? If there's microorganism in that tissue, guess what? Microorganism could also get in where? Lymphatic vessel. So the drainage is one direction. So all that will go to where? Lymph node. After lymph node filter it, eventually the fluid back to where? To the heart. You see? So now you see the function of lymph node. Before the interstitial fluid back into heart, lymph node will filter it, will check it, see if there's microorganism. Yes, question. Lymphatic drainage, and nowadays lymphatic drainage is a kind of fashion stuff. Like, oh, you less bloated, right? Like, you know. So it will help because the direction is one direction. They have walls to prevent backflow. So if you massage it, you can increase the lymphatic drainage, reduce edema. Yes, yes. So this is the way for microorganisms. If there's a microorganism in the tissue fluid, they can go into the blood capillary bed. That's a sepsis, right? They can also go to lymphatic drainage. That's once it passes through lymph node, that's the inflammation right there. Make sense? Now, any question about the lymphatic vessel? Yes? So eventually this is picture so the right side and the left side lymph fluid drainage. So the left side, including lower limb and the left arm, eventually goes to thoracic duct, drain into where? Left subclavian vein. And then the right side, the head, right side arm, you know, they go to what? Right subclavian vein. So after that, they go to the heart. You see? Make sense? So as it travel, guess what? This lymph fluid, as it travel towards the heart, see the lymph node checking it. So lymph node like checkpoint. Please listen, okay? Before you drive through like what's a border patrol agent? Walk to the heart. I need to check. Do you have, you know, whatever documents, right? You get an idea, right? Right? Because you don't want the microorganism to get in the heart, right? Otherwise, you know, carditis, all that stuff. Yes. Okay. Awesome. So over here, this is slide show you the detail. You see, this is capillary bed. So that's the beginning of lymphatic drainage. So they go through the lymph node. Once they go through the lymph node, they could go to the bigger vessel. Eventually, they dump into the vein system, back to the heart. See? Now, if you have inflammation in the lymphatic vessel, what's the symptom? Lymphangitis. Lymphatic vessel inflammation. Itis also inflammation. Inflammation in lymphatic vessel, lymphangitis. How do you know? That's a symptom. You see, lymphatic vessel, right? Inflammation. sporotrichosis. You guys remember sporotrichosis? I think last quarter we learned it, right? Rose-Gardner disease, see? Right? Inflammation on the lymphatic vessel. Yes. Another one we're gonna learn, another disease we're gonna learn in this quarter. Elephantiasis. So if you have a lymphatic drainage, reduce edema. If you have severe edema, what you have? You look like an elephant trunk for the lower limb. That's elephantiasis. What happened? What happened? It's worm infection. Worm infection in the lymphatic system. When the worm infection, you see worm big, they block the duct, so the fluid cannot come back. Severe edema. Right? Okay. Now, besides the lymphatic system, besides the cardiovascular system, the transport to the microorganisms, which structure also help us to grab the antigen? That would be what? Look at this picture called what? Reticular endothelial system. So reticular endothelial system, when you look at this picture, right? It's distributed, this connect tissue, connect everything, right? So loose connect tissue, reticular endothelial system, they also grab what? Antigen. Yes? Make sense? So what's the correct answer for this question? What's the correct answer for this question? All of this, yes? All of this, all of this, all of this. Now, I want you to look at this picture closely. What type of cell involved to grab antigen in the reticular endothelial system? What type of cell? You see the macrophage, neutrophil, dendritic cell, they draw it out, yeah? What type of cells? All of this. Does that make sense? Picture, we're solid words, right? Just look at each other. Makes sense? Yes? Okay, awesome. So, answers all of this. Yes? Now, let's look at this picture and answer that question. Look at that picture, right? You can see our, yeah. Which one? Thymus is the primary lymph organ. You see how many of thymus lymph organ? Yeah. Thymus is primary lymph organ. Yes. Rest of it is secondary lymph organ. Make sense? Right. Right answer is which one? Yeah. So, ready to move on to the next file? Cells of the lymphatic system. So the cells, right? And let's review the hematopoiesis again, right? The cells of the lymphatic system come from where? Come from where? Stem cells. Stem cells in where? Red bone marrow. Hematopoietic stem cell in red bone marrow, right? So cells renew, they replace themselves, and also they start differentiating into common lymphoid progenitor, common myeloid progenitor. So same information, different drawing, yes? Red blood cell come from which lineage? Red blood cell come from which lineage? Common myeloid progenitor. Make sense? A picture? You guys able to look at the picture? Yeah? Okay, awesome. So again, how do this, who should guide this process? Called what? Hematopoietin. That's the cytokines. So cytokines, growth hormones, hematopoietin, class-1 cytokines, they guide the hematopoietic stem cell by interaction with the receptor on the cell membrane to become which type cell. Let's review what we learned. Yes? Okay, awesome. So now, so here, this is again review, it's a different drawing review. So the red bone marrow, right? So that's where you have the hematopoietic stem cell, and the B cell will be stay there gradually, go to the school, pass the lactic selection, become, the B cell with the receptor. And then the T cell will go to the thymus gland, and then pass positive lactic selection, yes. Then after the graduation, they go to where? Go to the spleen, lymph node, the gall, mold, salt, all that. Yes? So let's review. So this is review. So T cell originating where? Originated in red bone marrow. The maturing where? In the thymus. So E and C, right? Yes? Make sense? Right? Did you learn? Now we're going to look at this one. How do we differentiate how T cell, B cell, the CD markers? How do we know? It's what type of clothes we wear, right? Reflecting on what? What type of protein on the cell membrane? Right? So have B cell receptor, T cell receptor. So this is the picture drawing the B cell receptor and T cell receptor. Yes? And then based on picture, can you answer that question? Right? Which one binds to antigen directly? The B cell receptor. The T cell receptor have to go through what? Major histocompatibility complex. Yes? So that's the difference between T cell receptor and B cell receptor. Follow so far, everyone? Correct answer for this one is which cell? T cells. And then we talk about CD4, CD8. If you have CD4, CD8, you probably have CD2, 3, CD20, CD19, etc. Yes? That means on the T cell, B cell, all the cells we have, they have more than one type of protein on the cell membrane. Yes? Make sense? So the CD4 is talking about this is the only receptor found on T helper cell. CD8 is this is the only receptor found for the cytotoxic T cell. Our cell don't have those receptors. That's why we use a unique CD marker to label the cell. That means the cell have multiple receptors. Yes, we just find the unique one. Oh, this is one our cell don't have. You the only one have that. We just use this receptor to name it. That's all. Got it? Okay, awesome. So now look at here. We have B cell, T cell, T helper cell, TC, cytotoxic T cell, natural killer cell. We have 4 cells. Look at the CD marker. It's not like this is all. That's just the CD marker is well studied for now. Do we know everything? We don't know everything. Okay, but we know a lot. Look at how many we know, right? So we're going to use this table to answer next question. Ask you guys to dig in through, okay? Epstein-Barr virus, HIV virus. How do they infect the cells in our immune system? You have to look at the table. You have to dig it. I want everyone to dig it. You don't have. So look at this CD marker, right? You see? So how about I give you the first one? You see CD21. CD21 only found in where? In the B cell. How the virus, this is a receptor for what? So the receptor complement system, also Epstein-Barr virus. So how do Epstein-Barr virus cause Burkitt's lymphoma? Because they can use the CD21 to get into the B cell. Yeah. Now you see that each CD marker have the function. And some virus sometimes take advantage of that CD marker get in. Right? So now you guys answer the next question. HIV, don't we learn HIV virus last quarter? So HIV virus gets into the T helper cells through what? CD4 receptor. So the answer is what?
How do you know?
CD4 plus what we learned last quarter. CD4 receptor. That's how HIV virus causes immunocompromised condition. And for the first one, Epstein-Barr virus, how do we know? It's right here, right? CD21. Make sense? CD21, so this answer is CD21, HIV is through CD4, right? So the correct answer will be what? C. Right? See the connection. Hopefully you last quarter still another one.
I mean, I understand because CD21 obviously because I couldn't see before, but by the table, how can I know?
By a table, this is how
The
you know.
CD4 because it's not mentioned.
Yeah, by a table, you see Epstein-Barr virus right here. And the CD4, this is the information have to be come from the last quarter's information, because we teach you last quarter, I just hopefully you remember. If you don't remember, now we remember again. Okay? Yeah, that's why we repeat ourselves, yes? So you kind of understand how the virus get in the cell. So use the spikes bind to the receptor on the host cell. And the host cell have receptor for something else, so they just take advantage of that getting. I like a review. Yes, right? Okay. You guys remember what we learned. Okay, so now I want you to answer this question based on those pictures. Based on this picture. So that way, I want you to get comfortable, just look at the pictures to figure out information. So based on that picture, what's the difference between naive and effector cell? So you see the naive T cell, B cells, and effector T cell, B cells. Yes? Can you see it? Right here? So what's the difference?
Vectors can go straight into the skin, whereas naive have to go to the spleen.
Yes, good job. Right? So naive T cell, B cell, they come from the bone marrow thymus. So naive T cell, B cell, they are the first graduate from the primary lymphoid organ. So kind of like when you finish graduation, you get your diploma, you have to look at the patient yet, you are naive chiropractor. Okay, but you get an idea what you call it, right? So bone marrow and thymus, we're talking about is a school for T cell, B cell, right? So when they graduated, they are naive because they haven't fight the war yet, yes? Right? And then once the naive go to the spleen lymph node, that's where they start to look at antigen. And then how do they leave? So when they leave the spleen lymph node, the effectors means, oh, I find antigen. They fit my profile. I'm going to go out there fight the war. So that's what effector T cell, B cell. So effector T cell, B cell already look at antigen. Right? They already look at antigen. They get activated. And naive T cell, B cell is they're ready, but they haven't looked at antigen yet. Make sense? So how do you know? Guess what? You can look at it on a microscope. Look at this picture. Right? So look at this picture. This is naive cell. So you have giant nucleus, not much going on. But once you get activated B cell, for example, so the plasma cell, effector state, see the shape change. So what's function of plasma cell? That's an effector cell. So the effector versus naive, you see the morphology difference? What's the function of plasma cell? Everyone? Produce antibody. How do they produce antibody? They need a long rough ER. You see the long cytoplasma space. You have rough ER, ready to do the work. Effector T cell, B cell, ready to do the work. Naive, they just a reservoir. Got it? Okay. Now, right, so again, now look at this picture, right? Look at this picture. Answer that question. Which cells could activate the naive T cell? That would be what? APC is antigen presentation cell. APC equal to what? Antigen presentation cell. We learned today which cells are antigen presentation cell. Dendritic cell. Yeah, there you go. Make sense? Okay. And once they get activated, you see naive CD8 becomes cytotoxic T cell. So effector cell, cytotoxic T cell. Naive CD4 become effector cell, could be T helper 1, T helper 2, T helper 17, inducible Treg, T helper 22, T helper 9, variety of T helper cell. Got it? Okay, awesome. Now let's look at the Treg. So T-reg, we're talking about taxes, right? T-reg, we call it from the IRS. So IRS, natural T-reg, they form in the thymus. So during the positive selection, negative selection, natural T-reg come from the government, from White House, natural T-reg. If you heard locally, called what? Inducible T-reg. So have two types. So regulation, you want to check. T-reg typically reduces inflammation, reduces immune response. So how do we check? So the natural T-reg come from thymus. Inducible T-reg actually in the lymph node, in the peripheral tissue. Hey, we won't do a local control, just fine-tune it. Right? Even in the local, you still have ability to fine-tune the immune response. Because the immune response only helps you if it's in the appropriate amount. Overactive, underactive immune response, that's disorder. So the T-reg could help you to regulate the degree of the immune response. Make sense? Right? So that means so our immune system always want to fine-tune it. Yes? Okay. So one of the protein, FOXP3, this is a protein in the nucleus. In the nucleus, once the FOXP3 in the nucleus get activated, then you become a T-reg. You not do work, you just regulate. Sounds good? So now, you guys have questions so far? Going good? Right? Over here, we're just going to look at the myeloid progenitor. So lymphoid progenitor, T cell, B cell, natural killer cell. Myeloid progenitor, besides regular cell, platelet, have a bunch of cells. You have neutrophil, macrophage, dendritic cell, mast cell, eosinophil, all come from what? Myeloid progenitor. And then the shape. Look at under microscope. Each cell has distinct shape. So you can do the blood work, you can count. Sometimes you do blood work, increase by the cell, decrease by the cell. Means how do you know? Because each cell group has distinct morphology, you can count them. Okay? Sounds good? So far okay, right? So now we know the variety of white cell and the function. Now I want to pick a couple of cells to look at it. First group cell I wanted to show you. It's called the granulocyte. So this is where we're going to remember. So everyone, who are the granulocyte? Everyone, who are the granulocyte? Neutrophil, eosinophil, basophil, mast cell. So who are the granulocyte? Everyone? Yes. So then the T cell, B cell, natural killer cell, we call them agranulocyte. Means we do not have granule. And granulocyte means if you look at the cell, the cytoplasm, a lot of dots, granules. So granulocytes, you got to remember, because we're going to test you in the midterm exam. You got to remember who are the granulocytes, who are the agranulocytes. Okay? Sounds good? Then, why do we have different types of granulocytes? Why do we just have one type? Then you look at what's the difference between neutrophil, eosinophil, basophil, mast cell. So this is the table show you what's inside the granule. So you see, inside each granulocyte have different contents, right? You can think of this granulocyte as a soldier. They have pocket, hold weapon. Neutrophil will be have pocket, carry the book bag with pocket, hold the sun. Eosinophil have pocket, hold knife. Okay, and the basophil have pocket, hold the gun. Right? You get the idea, right? Have different weapon system. Depending on what type of enemy you face, and you use certain weapons more suitable for that. Right? Now, we're going to use this slide to answer this question. So the answers in the previous slide, you go look at the slide. You look at the slide literally 5 minutes.
A?
Yeah, in the neutrophil. So defense lifeline is specifically effective get rid of bacterial infection. So we have increased neutrophil indicate bacterial infection. Yeah? See, I'm making connections, right? Now let's look at here. We have a lot of inflammation. Oh, allergy, a lot of inflammation, right? Histamine, leukotriene, which cell have those? Which one? Mast cell, right? Mast cell basophil. So you get idea, right? Increase inflammation. Now, natural killer cell, they are agranulocyte, but they can't kill the cell. So they do not use the preformed vector, but they have what? Porphyrin granzyme. So porphyrin from the name, porphyrin, they form in the hole on the membrane of the cell. Granzyme going in, cause cell death. So how do natural killer cell kill the cell? Through what? Porphyrin and granzymes. Yes? Okay. Now let's look at here. You guys remember phagocytosis? We learned last quarter. Second line defense, phagocytosis, right? Which cell carry out phagocytosis? Macrophage and neutrophil. Right? Macrophage, neutrophil, yes. Phagocytosis, yes. Okay. What the phagocytosis? This is review. We learned the last quarter. Phagocytosis, that's what it looks like, right? So we're talking about the granulocytes have preformed pocket, right? Now, another mechanism of phagocytosis, you can see it, macrophage and neutrophil. So step of phagosome formation, you have how do they kill the microorganism? Through the lysosome. Lysosome is what? Recycling center. Lysosome will digest the microorganisms. In addition, they also increase the reactive oxygen species through what? Respiratory burst. Respiratory burst, talking about the macrophage and neutrophil. After they carry out phagocytosis, they start to increase cellular respiration. You learn cellular respiration in biochem. So when you use oxygen, you produce what? A lot of reactive oxygen species. So you have a lot of reactive oxygen species plus the stuff inside lysosome. Get rid of it. That's phagocytosis. We do have an amazing video in week 1 to review that. Yes? Okay? So now macrophage is very important, right? So now I'm going to show you the function of macrophage. So macrophage, depending on the location, we have 2 groups. Wandering macrophage, fixed macrophage. Okay, so now I'm going to ask a question, you guys see if you can answer based on this slide. Where do you find the wandering macrophage? In the blood, wandering in the blood. Where do you find the fixed or tissue macrophage? Specific organs in where? In the brain, in the lung, in the kidney, in the spleen, liver. Yes, all the important organs. They have their own tissue macrophage. The kind of wandering macrophages like patrolling, state police. Fixed macrophage could be your local policeman. Yeah, because the immune system functions like police function, just to arrest, get rid of the thief, the murderers. That will be microorganisms coming and wait, right? Sending nutrients, they will get rid of it. Yes? Okay. So now, look at here. So some of the macrophages, depending on the location, have different names. For example, in the lung, called what? What's the macrophage in the lung? Alveolar macrophage. What's the name of macrophage in the brain? Microglia. What's the macrophage in the, under the skin? Langerhans cells. So you get an idea, right? Sometimes they have this specific name. Right? So now the next couple is just to show you that some microorganisms directly infect the monocyte or macrophage. Let's look at a couple of examples. Anaplasmosis. Anaplasmosis, which microorganisms cause anaplasmosis, everyone? Right? Anaplasma phagocytophilia. So anaplasma phagocytophilia, you can see it from the name. In fact, which cell? The cells involve phagocytosis. Right? How do you know the bacteria multiplying in the monocytes and macrophage? You see, in the cytoplasm, bacteria multiplying there. You see? Right? So this one, yeah, tuberculosis, that's alveolar macrophage, we're going to look at that. Yes, yes, anaplasmosis, right? Who's the bacteria?
Deer teeth.
Yes, yes, good job, good job. Tick bite. So tick bite, we learn transmitted Lyme disease and plasmosis. Yes. Another one is ehrlichiosis. You see ehrlichiosis, vector multiplying and monocytes, ehrlichiosis. So have different type of ehrlichiosis, different type of bacteria. Yes. Again, this is a tick bite. Another one, in fact, the monocyte macrophage is what? Protozoa, not bacteria. So ehrlichiosis, bacterial infection, visceral leishmaniasis called what? Klar-Azar, black fever disease. Have you heard of those diseases?
Oh, yeah.
Right. So the vector sandfly in South America, sandfly. So once this fly bite, the protozoa multiply inside macrophage, and the macrophage carry it to the visceral organ. That's yeah.
Ehrlichiosis is from tick.
Ehrlichiosis, yes. Over here, visceral leishmaniasis from sandfly. Yes. Right. You see, once the protozoa multiply in the visceral organ, you see, how do they get visceral organ? Sandfly. Because they use the macrophage, monitoring macrophage carry it to visceral organ. Yeah. Another one is called what? Chagas disease. Chagas disease, kiss and bug. Kiss and bug. Why you call it kiss and bug? Like to kiss you, bite your face, kissing you. Kiss and bug. But once they go in, they can use the macrophage, the multiply inside macrophage. Macrophage carry a microorganism all the way to the visceral organ. Enlarge heart, megacoli, enlarge esophagus. Yes. How about that? And you guys heard of tuberculosis, multiplying inside of your macrophage. Right? You like that story? Okay. Awesome. All right. Now, let's look at here, review. This one, all of this. Yes, how many of the microorganisms? Yeah, all of those. Good job. Yeah. Right. So with the dendritic cell, what's the function of dendritic cell? Antigen presentation. So the shape of dendritic cell, you can see the shape. It has a lot of ruffling, tentacle-like structure. So it's not smooth cell membrane, it's ruffling. So this shape allows them to grab anything. Yes? Right? So based on this, we can answer the next question. Dendritic cell come from where? All of this we reviewed that. Yeah, good job. Now, another one, heard of malaria? Plasmodium. So this is the protozoa going inside where? Red blood cell. When they go inside red blood cell, we call them what? Antigenic concealment. What's concealment in English? Concealment means what? Hide. So why they hide in red blood cell? Because red blood cell, once you go in there, red blood cell don't talk to T cell, B cell. So when you have infection, nobody come. Yes. Okay. So that's the right. So that's the same for microorganisms. If you manage to hide inside red blood cell, nobody come. Okay. So that's a quantity infection. Now, based on previous slide, answer this question. Which one? Which one called what? Plasmodium falciparum. Plasmodium. So how do they escape host immune system? Antigenic concealment. So which one is correct answer for this question? Yeah, there you go. You like that story? Yeah, now you want to check the knowledge check.